All right, so in lesson 7, we are going to talk about antivirals. So we're going to be talking about influenza, HIV, and AIDS. Here are your understandings and applications and skills. So let's talk about viruses. So viruses are submicroscopic and can only be studied with an electron microscope. They come in all shapes and sizes. They are simple and small structures, and there is debate about whether they can even be classified as living organisms in their own right. They contain only two main components, protein and nucleic acid, which includes either RNA or DNA. They have no cellular structure and are only capable of reproducing inside another living cell. A little bit about the structure of a virus. Viruses have a core consisting of their genetic information, which is carried in the form of that DNA or RNA, and it is surrounded by a protein coat known as a capsid. This capsid consists of identical protein subunits called capsomeres, and its role is to protect the genetic information in the core. Together, the capsid and the genetic material is called a nucleocapsid. Some viruses, such as the human immunodeficiency virus, otherwise known as HIV, also have a lipid envelope that surrounds the nucleocapsid. All right, so here we can see a picture of some viruses. On the left, we have what is known as a T4 bacteriophage. So you see the DNA inside the protein coat. And then on the right, we see an example of an HIV virus, which is made up of viral DNA inside its coat of core proteins and the lipid envelope, as well as other viral proteins. So viruses take over the function of another cell, which is known as the host cell, and then they use that other cell to carry on reproduction. One of the things you have to be aware of are some of the differences between bacteria and viruses, which you can see in the table there below. All right, the big thing is that viruses are not living. They do not have a cellular structure, and they need a host cell to reproduce. Okay, because of all those reasons, that is why viruses are considered not living, whereas bacteria are. So, how does the body then fight back against viruses if you are infected? Well, the body's defense system usually responds to viral infections by producing specific antibodies, which act against a virus in the immune response. This often leads to protection, known as immunity, against repeated infections with the same virus. But sometimes the virus is not completely eradicated from the body and remains dormant in cells. This can cause a flare up on another occasion, such as some herpes infections, which cause cold sores. So, when you are infected with a virus and your body produces these antibodies, okay, you then have an immunity against that virus. One common example is chickenpox. Somebody who has had the chickenpox when they were younger, they are immune to getting it again later in life. Viruses cause many deaths, right? Such as the flu, measles, meningitis, polio, HIV, Ebola, avian flu. We can now add the coronavirus. Okay, viruses are definitely something that we need to take seriously because it is a difficult fight. Treating viral infections is particularly difficult because the viruses live within the host cells and so cannot be easily targeted. They need those host cells to reproduce. So whereas with bacteria we can use antibiotics such as penicillin, we talked about that. Well, penicillin, for example, can target a structure such as a cell wall, but there is no viral structure. There is no structure to target. Another problem is the speed at which viruses can multiply, so that they are often spread through the organism by the time that symptoms appear. They can reproduce very quickly once you are infected. In addition, virus particles have a tendency to mutate rapidly, which means that they make small changes in their genetic material, and this changes their susceptibility to drugs. So these mutations, which can occur very quickly, the drugs that we use are not as effective. One of the ways we can target these viruses are with vaccines. So vaccines were first introduced in the 18th century and today are a major aspect of preventative healthcare known as prophylactic treatment. Vaccines work by stimulating the body to prepare specific antibodies which can give immunity. Successful vaccination programs have reduced the incidence of diseases such as cholera, polio, and measles. Unfortunately, there is some limitation to vaccines. One of the questions is, why do we need a flu shot every year? Well, from before, viruses mutate. So the vaccine that we have this year is not always as effective, and it may change year to year. In addition, sometimes our immunity wanes as we age, so we need booster shots to ensure continued protection. All right, when you all were younger, you got shots, you got vaccines, but you had to then go back later on to get these booster shots so that you are fully covered. If there is no vaccine for a specific virus or the current vaccine is not effective, we have to rely on antiviral drugs to help us treat these infections. So how in general do antivirals work? Okay, well, different ways, different methods. Some will alter the genetic material within the cells. Once inside a cell, the drug is converted into an active metabolite that becomes incorporated into the growing DNA strand, which is needed for viral replication, halting its synthesis. For example of a drug that acts in this way is acyclovir, which is used to treat cold sores. It stops the viral DNA replication and so stops the virus from multiplying. Okay, so if you target the DNA or RNA and can prevent the multiplication, then you can stop the virus. Some will inhibit the activity of enzymes within the host cell that are necessary for the formation of new viruses. An example is indinavir, which is used in AIDS treatment. It inhibits the HIV enzyme protease, which is essential to the assembly of functional new HIV viruses. Okay, so targeting enzymes that can be used in the production of new viruses. Some stop the viruses from infecting host cells by preventing them from binding to the host cell surface and gaining access into the cell. Some drugs used to treat AIDS work in this way. Okay, because remember, if a virus can't bind to the host cell, then it can't replicate itself. Some prevent the virus from leaving the host cell so that it cannot infect other cells. For example, two of the drugs that we're going to be talking about, oseltamivir and zanamivir. The flu Influenza, we usually do not think of getting the flu as a death sentence, but roughly 500,000 people each year die to the flu. And infectious disease doctors are constantly on the lookout for a mutation that will cause a pandemic, a global outbreak with a high risk of casualties. There are actually two types of influenza, or flu. Flu is caused by two different types of viruses, influenza A and influenza B. They are spherical viruses and they have RNA as their genetic material. These viruses have specific proteins on their surface, of which two play a key role in their life cycle. So the two flu proteins that are on the surface include hemagglutinin, which is given by the letter H. This is a glycoprotein that enables the viral particle to dock with the host cell before it enters. And the other protein is neuraminidase, given by the letter N. It is an enzyme that catalyzes a cleavage reaction so that it can break off. This allows new viral particles to escape from the host cell and spread infection. The enzyme snips off a type of sugar molecule, sialic acid, from glycoproteins on the surface of the host cell membrane. Okay, I'm going to show you a picture of this on the next slide. The naming system of viruses, such as H1N1 and H5N1, refers to the different forms of these molecules that are present. H1N1, if you all remember, about 10 years ago, was the avian flu or the swine flu. All right, so here is an example of a viral cell with the hemagglutinin and the neuraminidase on the surface. You can see how it will bind to a host cell via the hemagglutinin. It'll replicate its DNA or RNA, and then it will release via the neuraminidase. So if we can prevent one of these actions from taking place, then you may be able to prevent the spread of that virus. Okay, if you can change the life cycle of the virus. Well, of these two, the hemagglutinin and the neuraminidase, neuraminidase has been the better target for drug design, and so it has become the focus of research. So neuraminidase binds to its reactant sialic acid, which is the substrate, at a specific region known as the active site. It is the binding between the enzyme and the substrate that gives the catalytic action, as it provides a reaction pathway of lower activation energy. Chemicals that interfere with this binding are called inhibitors and usually have a specific interaction with the enzyme. So let's talk about the drugs that have been used to target this neuraminidase. So the 3D structure of neuraminidase became known through X-ray crystallography in the early 1990s, and it included details on its active site. So by knowing this, researchers were able to design molecules which could bind at the active site and so block the binding of substrate and act as an inhibitor. So the first neuraminidase inhibitors were designed by a team in Australia and led to the production of zanamivir, which is commonly known as Relenza. And this was approved for use in the year 2000, and it was closely followed by the production of oseltamivir, which is commonly known as Tamiflu. Okay, so on the next couple slides, we're going to take a look at some structures of the site of the sialic acid on the host cell, as well as the drugs themselves. So here we have the sialic acid, okay, with some of the functional groups highlighted, right? This is the structure that the neuraminidase on the virus attaches to. So here are the two drugs which are commonly used for the flu. Okay, we have Tamiflu and Relenza. And again, you could see the functional groups, which are highlighted and colored there. You could see that they are administered differently. Tamiflu is given orally. Relenza is inhaled. Relenza has no resistance reported, although Tamiflu does have some resistance to some rare strains of the flu virus, and there are some side effects, as you can see there. Okay, you can see how the structures of these two drugs correspond pretty closely to the sialic acid from the previous slide as well. Okay, so in these last couple slides, we're going to talk about HIV and AIDS. So AIDS was first recognized in 1981 and was found to be caused by the HIV virus a few years later. Nowadays, there are believed to be more than 34 million people infected with HIV worldwide, and approximately 2 million deaths occur each year from AIDS. Okay, so a very serious disease, and a lot of action has been taken to fight this. So let's talk about the HIV virus. So the HIV will primarily infect vital white blood cells in the immune system. These cells are called CD4 T cells. So the HIV virus will bind to a specific receptor protein on the cell surface and then penetrate the cell. Now, HIV as a virus is known as a retrovirus, which means that its genetic material is in the form of RNA rather than DNA. So what happens is the virus will release its RNA into the cell, and the enzyme reverse transcriptase controls the synthesis of viral DNA from this RNA. The viral DNA integrates into the cell's own DNA and replicates with it when the cell divides. Viral particles are produced within the host cell and are released in large numbers when the cell dies. Okay, so we're going to take a look at how this happens right here. So you can see here's a computer image of what is going on. So if you follow along, it says that the viral particles shown in green surround the white blood cell shown in blue and attach to its surface using specific proteins for recognition. Viral RNA, shown in pink, is then injected into the cell and using reverse transcriptase synthesizes DNA, which integrates into the host chromosome. This can be seen in the white cell nucleus in the center. New viral particles are assembled within the cell and are shown at the bottom, budding from the cell, taking part of the membrane as an envelope. Okay, so we can see how a viral HIV particle can attach to a white blood cell. It will inject its viral RNA into the host cell, and then this RNA will be injected into the nucleus after it has been synthesized into DNA using that reverse transcriptase. Okay, and once it is in the center here, okay, it will then be released as new viral particles, which can then go off and find other white blood cells, and this process just repeats itself. So why is HIV hard to combat? So the virus will destroy helper T cells, the very cells in the immune system that should be defending the body against the virus. The virus tends to mutate very rapidly even within a patient. it is thought that there is more variation in HIV in a single patient than in the influenza virus worldwide in a year. These variations means that the virus escapes the immune response, so the patient has to make a response to the new virus, and the virus often lies dormant within host cells, so the immune system has nothing to respond to. Okay, so because of these three actions, HIV is really hard to target, and it is an issue that continues to this day. But there are some drugs that have been used to help target HIV. These are known as antiretrovirals. So these drugs target and interrupt the following different stages in the HIV life cycle. So it'll interrupt binding and fusion of the virus to the receptor on the CD4 cell membrane. Drugs target reverse transcription of viral RNA to DNA. Drugs have been used to prevent integration of viral DNA into the host chromosome. And finally, releasing the new viral particles by budding from the host cell surface. Okay, so antiretrovirals, these are the drugs that are used to combat HIV. Of these targets, inhibitors of the viral enzyme reverse transcriptase are the most widespread. They include drugs such as ACT, also known as zidovudine, which was the first antiretroviral drug to be approved. Now, rather than taking a single drug, it has been found that the best results occur when a combination of different antiretrovirals are used. So they could include those reverse transcriptase inhibitors plus a third drug. These can all be combined into a single pill once daily, and it's often been referred to as a triple cocktail. Unfortunately, the cost is one of the worst things about antiretroviral drugs, and per patient it costs about $12,000 per year. Now, there has been ongoing intense research about developing a vaccine for HIV and AIDS. There's hope that you may be able to control the infection in people who are HIV positive. But for those who are HIV negative, that has not been possible to provide a vaccine to provide immunity. This is because viruses mutate, and the fact that the immune response seems to act too slowly in the case of HIV infection. Okay, so we got a couple questions here to consider. Number 16 says, why are viral infections not able to be treated with antibiotics? Okay, so we talked about bacteria versus viruses earlier. And we know that viruses lack a cell structure, and so they are difficult to target. Antibiotics specifically will interfere with bacterial cell walls, and so we can target a specific structure, whereas viruses, they need a host cell to replicate, and therefore we don't want to target those healthy cells. Number 17 says, with reference to the structure of the influenza virus, explain why it is possible to suffer from flu several times during a lifetime. Okay, so we're talking about the structure of the flu virus itself. Okay, so that was the H1N1 or the H5N1. So that deals with the hemagglutinin and the neuraminidase. So because we have all of these different combinations and mutations and mix and matching going on, there are different strains of the flu. So the flu that you have at one point in your lifetime is and probably will be different than another one. So therefore, the body's immune response is going to be different to each of those influenza. There is no immunity to the influenza virus. Number 18, explain why the antivirals Tamiflu and Relenza must be taken within a very short time after the appearance of the symptoms of flu. Okay, so if you think about all viruses and specifically the flu virus, what has to happen is they must attack a host cell so that they can replicate and be released so they can infect other cells. The life cycle of the virus in a nutshell. So, Tamiflu and Relenza do not prevent the flu virus from entering cells, but they act to stop it from being released. So, if you do not target it early, too many new viral particles may have already been released. And last but not least, number 19, discuss some of the challenges and successes in the global response to the AIDS pandemic. Okay, HIV and AIDS has been around for 30 plus years, and there have been some challenges and successes in trying to find a drug or a vaccine to this virus. So some of the challenges include the cost of the drugs, distribution and availability, patient compliance to regimen and multiple drug treatments. There has been sociocultural issues related to being HIV positive. There are, however, a great deal of successes. So there's new and more effective antivirals that can be used in combination, so that triple cocktail. There is better screening of HIV positive patients, and using drugs has been better as we've learned more information about the virus.